produce look like just little water things swimming. Morning, at least it's morning for me, so wherever you are, hello. So today I'm going to do the infection assays, that is applying the fungus slash the pathogen to the plants. So I'm going to show you what they look like, because remember in the inoculum initiation video I was just applying the fungus from those small vials that looked like it was just water onto the plate, and there was nothing on the plate when I was done. So at 7 day, I'll show you what they look like. That looked like they didn't have anything on them at the time of inoculation. They have things growing on them. Once I'm in the BSC, I'll give you a better look at it, but it's peachish, pinkish, and that's what the pathogen grows like on nutrient-rich agar. It grows like yeast-like cells, and so that is great. I don't see any signs of contamination. It's just one type, so that is good, and I'm excited. So we'll get started. This is a spectrophotometer, and I'm going to use it to quantify the inoculum to have an idea of how much of cells are in the inoculum once I make it up. And it takes a while to warm up or to turn on and come up to the right settings. So another thing I like to do when I come in, if I know I'm going to use it, is to turn it on. Okay, I think it's unplugged, so let's plug it in. Because I'll be using the BSC to prepare the inoculum, what I like to do is to get it up and running, because it's going to take a while, maybe 15 minutes to do the sterilization of the BSC. So I'm going to do that before I do anything else. Doing its thing, so the room is about to be really loud. But anyway, while that's happening, I'm going to go gather some things I need. I'm just going to grab all the things that I need for today, that I need in the BSC. So the plates. These are important bits. Oh, that's what they look like. Can't really see from here because of a condensation, but I need the plates. So I inoculated 12 of them. So I have them here. I am going to make up 1 liter, 1000 ml. So I have a 1 liter Erlenmeyer flask, sterilized funnel, because I'm going to filter the spore suspension to remove any large pieces of maybe hyphae or any debris like pieces of agar, because I might gouge the agar when I'm scraping them. Sterile cheesecloth, again for the filtering. Water. So I'm going to use about 1000, and I need also an empty, I might need an empty bottle, not sure yet, seeing that I have the Erlenmeyer flask, but I'm just going to grab one just in case. Empty bottle, and I already have the 1000 microliters or 1 ml micropipettes, so that is fine. What am I missing? Oh, I'm going to use a piece of black cloth, because I like taking photo of what the plates look like at the time of inoculation, of making up the inoculum. And yeah, I think that's it for right now, and we'll just go inside and work. Spray everything down with 70% ethanol, and then slowly put them into the BSC. And once I'm inside, you will see me prepare the inoculum. So essentially I'm going to add a certain volume of water to the plates, harvest the spores by gently scraping the surface of the plate, filter it, keep doing that until I have the 1000 ml. And once that's done, I will QC. So as a quality control, I like to stain about 10 or 20 microliters of the inoculum with lactophenol blue. Again, remember that stains a particular compound, chitin, that's in the fungal cell wall blue, and that helps to provide contrast, because if not, you will see, and they'll just look like just little water things swimming, but it gives it a blue tint. And again, chitin is one way of distinguishing bacteria from fungus. And again, chitin is in the exoskeleton of crustaceans, crabs, shrimp, lobster, all those things people enjoy eating. And then once that's done and I do the microscope part of it, just do it for the QC, I will test using the spectrophotometer to see in terms of OD units, using the electromagnetic wave to get the concentration. So if you did chemistry or maybe microbiology, remember this from it. This one might be different. This one is probably a bit older than the ones they have in universities. But yeah, that's about So this is a blank with the water, and this is my sample, and these are disposable cuvette. So I'll throw them out once I'm done, but usually in like a research lab, you'll have a quartz cuvette, and those are expensive and those are reusable. But when using cuvettes, you want to wear gloves, and then you want to wipe the outside of the cuvette, because your hands have oils, fats, your DNA, and then that will get onto the cuvette, and then you'll get a false reading. And so yeah, you just wipe, oops, well, I close it with the perfume, but you wipe the outside, and there is a, well, the edge is covered, but there's an upside down triangle, and that's so you know which direction to put it in. This is what it looks like. So I'm going to blank the machine with water. Remember, it's at 595. I'll press absorbance. and 00. So I'll just take the blank out and then put my sample in and then take the reading. That's the sample. Came from that. Put it in. And the reading is 0.574, so that's absorbance. I'm going to set up the sample so that I can do the QC on it, so that is looking at the relatively abundance and the shape and size of the spores or cells under the microscope. Then once that's done, then I'm finished with that part of it. So these tips are they fit the 2 to 200 microliter pipette, and they're non-sterile because they're open outside, and I'm just doing it outside, so this doesn't have to be sterilized. I have the frosted glass slide, the cover slip, and the lactophenol blue to provide contrast. And then this is a sample from the spectrophotometer that I did a concentration of. So I'll do that. There's a frosted edge of the slide, and I'm just going to use some lens paper to clean the slide. It's already set to 20 microliters. I like using the tips in order. Some people do them randomly. Okay, one is missing from there, but I like using it in order, my order. Then I just flip it to shake, move the parafilm because it's set, so you want to go to the first stop, straight on, slowly pull it up. My 20 microliters, it's hard to see because it looks like water. Slowly aspirate all the way to the last stop to get everything out. Release my tip. I'll grab another tip for the stain or the dye, the lactophenol blue. It's roughly the same volume, 20. Is blue. Then I'll prepare it up and down to give it a mix. Release that, and I'll add a cover slip because I'm going to throw away the slide once I'm done. I'm not going to pay attention to how I'm putting on the cover slip. I'm just checking to see that it's free from debris or well, there shouldn't be any fingerprint because I'm wearing gloves. Okay, just put the lens paper isn't smooth, so it leaves fuzz on it, and then I'll apply it like so, let it sit a bit, and then I go look at it under the microscope. At times 40, you're really starting to see distinguishable features. Ooh, look at them. So I added oil immersion to the slide so I can see at times 100. And look at these babies. Scope, when you're done, you wipe it down, you turn it off, the light, because you'll blow the bulb, and then you leave it on the lowest magnification. So you put it back at times 4, and then you lower, okay, the stage, you lower the stage. So yeah, I'm done.